Hello, class. Today we're going to go ahead and go over the structure and function of DNA and protein synthesis. In your textbook, it's under Chapter 12, the molecular biology of the gene. Molecular biology of the gene. All right, the learning objectives which we see set forth by the College Board are that you should be able to describe the structures involved in passing hereditary information from one generation to the next. And you should be able to describe the characteristics of DNA that allow it to be used as the hereditary material. Here's a video if you guys would like to start off with looking at. It's called the double helix, and it does go over the discovery of DNA. I know many of you guys have watched this video before, but if you haven't watched it or if you'd like to refresh your memory on the double helix, this is a really good video showing the discovery. All right, a little bit of scientific history. If there's anything you need to know, it should be this slide. It talks about the history of how DNA was discovered. So we just finished learning about genetics. And as we learned in genetics, we knew that chromosomes were being passed from one cell to another. We know the genetic information. But at that time, Mendel didn't understand that it was DNA that was actually being passed from one organism to the next. So the march to understanding that DNA is the genetic material started in 1908. And in 1908, T.H. Morgan explained or founded that genes are located on chromosomes. So remember, as we said, genes are sections that code for a trait which are found on a different chromosome. In 1928, Frederick Griffith described the transforming factor can change the phenotype. So whatever is being passed from one organism to the next can change the phenotype or can change the trait. In 1944, Avery, McCartney, and McLeod described that the transforming factor is DNA. So notice in 1944, what we're saying here at this point is that the molecule that's being passed is DNA. In 1947, 3 years later, Erwin Chargaff comes up with Chargaff's rules. A bonds with T and C bonds with G. 1952, Hershey and Chase discovered and confirmed that DNA was the genetic material that was being passed. In 1953, Watson and Crick determined the double helix structure of DNA. And in 1958, Nielsen and Stahl discovered the semi-conservative replication. Each one of these advances could not have occurred without the experiments beforehand. So they all built upon each other to find the discovery and the structure of DNA. Here's a brief video on Griffith's experiment. Please make sure that you take a look at this video. I know the AP exam likes to give some questions regarding Griffith's experiment, so please make sure to watch this video. And here is a small illustration of what Griffith experiment occurred using Griffith's transformation experiment. All right, so DNA. DNA, as we remember, is deoxyribonucleic acid. DNA is composed of 4 types of nucleotides. So DNA is made up of a long chain of nucleotides. Each nucleotide has 3 parts. It has the phosphate group, the deoxyribose sugar group, and the nitrogen-containing base. You should have remembered this from first semester, hopefully. Nucleotides always pair in the same way. So the base pair rules show how nucleotides always pair up in DNA. So remember, A pairs with T, or apple trees, and C pairs with G, Curious George, or cars in the garage. All right, Chargaff's rules. So Chargaff's rules stated that A bonds with T and C bonds with G. In each species, there is an equal amount of adenines and thymines and guanines and cytosines. Let's go take a look at these videos.
The Cavendish shop was to build us some tin models, and that took too long. And, finally in desperation, I made some out of cardboard.
So as we see, he's building his
I began
molecule.
moving them around. And I wanted an arrangement, where I had a big and a small molecule. And so how did you do it? Somehow you had to form link bonds. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh, so then I went to the pair and wanted this nitrogen to point to this one. I went like this. Whoa, they look the same. You can put one right on top of the other.
Now this here is him visualizing the building of DNA.
We knew he could just, even if we go up to the ceiling, we were building a tiny fraction of a molecule. Hundreds of millions of these base pairs in one molecule, all fitting into this wonderful symmetry which we saw, the morning of February 28th, 1953.
All right, let's take a look here at Chargaff's ratios, at the ratios. So as we saw in the brief video, we did see Watson talking about how they visualized and put together the structure of DNA using the base pair rules. Oops, let's go back. Sorry about that.
The DNA molecule is made up of very long chains of the four bases A, C, G, and T. In 1950, Erwin Chargaff published a paper stating that in DNA of any given species, the ratio of adenine to thymine is equal, as is the ratio of cytosine to guanine. This is known as Chargaff's ratios, and it was a crucial clue that helped solve the structure of DNA. Chargaff's ratios are universal. All forms of life obey this rule. Only the balance of AT pairs and CG pairs varies between species.
Okay, and as we could see here in this illustration, we notice that the nitrogen bases are held together by hydrogen bonds in the middle. And if you recall, on the outside, it's held by covalent bonds that are strong. So we have weak bonds located in the middle, and we have strong bonds, which are located on the outside. All right, there's two kinds of bases in DNA that you need to be aware of. There are pyrimidines and purines. So pyrimidines are a single ring base. So that would include thiamine and cytosine. They are pyrimidines. And then we have purines, which are double ring bases. And that includes adenine and guanine. Now, because the pyrimidine, which is a single ring, pairs with the purine, which is a double ring, the helix has a uniform width. So it has somewhat of a uniform width. So the single ring always bonds with a double ring base. The backbone, as we stated, is connected by covalent bonds, which are strong. Okay. So as we see here, our covalent bonds are located between the sugar and the phosphate group. Okay. And then we have our hydrogen bonds, which are located in the middle. And these here are weak bonds. So we want these bonds to be weak so that when DNA unzips, not a lot of energy is used to unzipping these hydrogen bonds. Okay. All right. So the hydrogen bonds are weak, which allow for DNA to wind for replication, as we stated once again. All right, the genetic material could not have been found without the X-ray from Rosalind Franklin. So X-ray diffraction, let's look a little bit about that. So Rosalind Franklin was a scientist who studied the structure of DNA using X-rays. Now she found that if a concentrated viscous solution of DNA is made, it can be separated into fibers. Okay, so it could be separated into fibers. Now, under the right conditions, the fibers can produce an X-ray diffraction pattern. She produced X-ray diffraction photographs, And this provided evidence that DNA had the following features: that it's a helix, meaning like it's a spiral shape, and that some portion of the helix is repeated, which as we see in this picture here. Watson and Crick's discovery built on the work of Rosalind Franklin and Erwin Chargaff. Now, Franklin's X-ray image suggested that DNA was a double helix of even width, as we see here. So this is what the X-ray would look like. Her photo is an infamous photo. It's called Photo 51. And like we said, without this photo, Watson and Crick would not have been able to discover DNA, or the structure of DNA, as I should say. Here's a video on Rosalind Franklin. Please go ahead and watch this on your own. Should be interesting. All right, Watson and Crick determined that, we said, the three-dimensional structure of DNA by building models. Just as we've seen in the video prior, this is the actual structure of the model that they built, okay? They realized that DNA is a double helix that is made up of a sugar phosphate backbone on the outside with bases on the inside. Same thing, this is the model which we've seen. All right, in the next section for our learning objective, the AP also wants us to be able to describe the mechanisms by which genetic information is copied for transmission between generations, okay, for transmissions between generations. All right, here's a very quick introductory video using the Amoeba Sisters on replication. Please watch this. It does a really good job of kind of explaining replication in case you get stuck. All right, DNA replication is the process of copying a DNA molecule. Please don't forget that replication occurs during the S phase of interphase, okay, during mitosis. Semi-conservative replication indicates that each strand of the original double helix serves as a template for a new strand in the daughter molecule. So as we know, DNA replicates is known as semi-conservative replication, semi-conservative replication. So as we see here in this illustration, we can see that the DNA is unzipping and that it keeps an old strand and builds a new strand. So it has an old strand and it builds a new strand. All right, replication of DNA. Replication requires the following steps: unwinding or separation of the two strands of the parental DNA molecule. So first of all, we have to separate the DNA. Then we have to base pair. So we add complementary base pairs between a new nucleotide and a nucleotide on the template strand. We join the nucleotides to form the new strand. Each daughter DNA molecule contains one old and one new strand. However, in prokaryotic replication, it's just a tad different, So because prokaryotes do not have a nucleus, they have a different replication. So bacteria have a singular circular loop of DNA known as a plasmid. So replication moves around the circular DNA molecule in both directions, and it produces two identical circles or two identical plasmids. The process begins at the origin of replication point. Now, in bacteria replication, it usually takes about 40 minutes, but the cell divides about every 20 minutes. And there's a new round of replication can begin before the previous round is completed. Because there's no nucleus, replication basically with bacteria or with our prokaryotic organisms has a high rate of mutations. There's a high rate of mutations. There's really not a whole lot of checkpoints. Whereas in eukaryotic replication, DNA replication begins at numerous points along each linear chromosome. So the DNA is going to unwind and unzip into two strands. Now, for each old strand of DNA serves as a template for a new strand. It's known as semi-conservative replication. So it keeps one old and it builds one new. The complementary base pairing forms a new strand paired with an old strand, and it requires the enzyme called DNA polymerase. So DNA polymerase. All right, eukaryotic replication. As we said, the replication bubble spreads bidirectionally until they meet. The complementary nucleotides are joined to form new strands, and each daughter DNA molecule contains an old strand and a new strand. So the directionality of DNA, you need to number the carbons, it matters. So when we look at the directionality of DNA, it usually runs from a 5', 3'. What does that mean? 5 to 3. So if I look here at my sugar, here would be our carbon 1, carbon 2, 3, 4, and our 5th carbon. Okay. Now, it runs anti-parallel. So the nucleotides and DNA backbones are bonded from the phosphate to the sugar between the 3' and the 5' carbs. So DNA molecule has a direction. Complementary strands run in opposite direction. So when you get this on the AP exam, you will probably see a strand, it'll say 5' ACGT3. On the opposite end is going to be 3, 5. So now we're going to do the base pairs. So A goes with T, G, C, and A. Okay, it runs anti-parallel. So what goes one way is the same thing going backwards. It's like the word race car. If I were to spell race car backwards, it's race car. Your DNA runs the same way. What runs one way is the same that runs the opposite directions. So we go 5, 3, and 3, 5. As we see, here's our bonding in DNA, same thing. We have our hydrogen bonds located in the middle, which are weak, whereas the outside is held with our covalent bonds, which are strong. So the first step in replication is we unwind the DNA and we use helicase. So helicase, anything that ends in ASE, is an enzyme. Helicase is the enzyme that unzips DNA and it unwinds DNA. Once DNA unwinds, we then add nucleotides using the enzyme DNA polymerase III. So DNA polymerase III is going to add the matching pair to each nucleotide. So to each nitrogen base, we add in the nucleotide sequence that is matching. Don't forget, A bonds with T and C bonds with G. So we add the bases, and you can only add nucleotides from the 3' end of the growing DNA strand. Okay, so the strand only grows from our 5' to our 3'. So 5, 3, and that's going to be our 3, 5. All right, so we also have in replication a leading and a lagging strand, Okay, this helps us to save time. So there are some limits of DNA polymerase III when it adds the nucleotide bases. It can only build on the 3' end of the existing DNA strand. Well, what about the opposite strand? Okay, on the opposite strand, so we have one that's building towards the fork. So as we see here, here is our helicase enzyme. So that's helicase. And as we see, helicase is unzipping our DNA. So helicase is unzipping the DNA. Then we have our DNA polymerase, which is building towards this area, which is called our replication fork. So it's going to add one nucleotide at a time, A, T, T, C, G, G, and so forth. Okay. Now, here's our original strand, and then here is the new strand, right? So this is called our leading strand. It's our leading strand because it's going to lead towards the replication fork. While the opposite strand is called our lagging strand, And what our lagging strand adds is what are known as Okazaki fragments. So these here are multiple nucleotide segments located together, okay? So there's like these large little fragments of DNA, okay? And these bases are going to match up with our base pairs here. So these are called our Okazaki fragments. Now, once our Okazaki fragments are built here, they are then glued together with this enzyme called ligase. So ligase is the glue. Ligase is the glue, and it's going to put these two molecules together. Okay, it's going to put those two molecules together. So one is building towards the fork and one is building away from the fork. One is building towards the fork and one is building away from the fork. So as we see here, here's our replication bubble. That's what we were talking about. So these replication bubbles occur at multiple strands on our chromosome. So here's our DNA, right? When I unzip our DNA, I have my leading strand and I have my lagging strand. So one's building towards the fork and one's building away from the fork. Now, there are some limits, as we see, of our DNA polymerase III. It can only build onto the 3' end of the existing DNA strand here, okay? We have a primer, which is called our RNA primase. And what it does is it serves as a starting sequence. So this is here going to be the start of us reading this segment here and building. DNA polymerase I removes sections of RNA primer and replaces them with nucleotides. So once our lagging strand is starting to be built, so here's our Okazaki fragments, this here then is glued together with ligase. Any gap or missing nucleotides is then replaced with DNA polymerase I. So every once in a while, the ends of the chromosomes start to erode. So all DNA polymerase can add to the 3' end of the existing DNA strand. The loss of the bases at the 5' ends in every replication. So sometimes the chromosomes get shorter with each replication, which then limits the numbers of cell divisions. Okay. They form what are known as telomeres. So at the end of your chromosome, we have non-coding sequences of DNA, meaning that this is sequences of DNA that has no code for absolutely nothing. It's just filler space, like AA, TT, GG, CC. Maybe these code for absolutely nothing, and it serves as a protective cap, okay? Telomerase is an enzyme that extends the telomeres, and it can add DNA bases at the 5' end, okay? So it's different level of activity in different cells. We see these telomerase enzymes high in stem cells and cancers. Why? Because we have high rates of replication when we look at cancer cells and stem cells. All right, so once again, let's go ahead and read this illustration. We have helicase, and as we said, helicase is unzipping our DNA. We have our leading strand, which is building towards our helicase or towards our replication fork, and it builds from the 3' to the 5' end. Then going away from the fork is the lagging strand, okay? We then add these segments of DNA known as Okazaki fragments, or these segments of nucleotides. The Okazaki fragments are put together and glued together using ligase. And then finally, DNA polymerase III is going to add any nucleotides that have been missing. And that there is how we build our DNA. All right, so the DNA polymerases. So you have DNA polymerase III, which is the main DNA builder. That adds a nucleotide. DNA polymerase I is responsible for editing, repair, and primal removal. So if we get a wrong base pair sequence, it will edit and fix it. So our bodies are very unique in the fact that we want to make sure that replication always stays the same. If our DNA replication is not the same, then we can have mutations, and that's how mutations occur. All right, let's take a quick look at this video together.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules.
So let's take a look real quick. As I can see here, this here is our helicase enzyme. And then I have one segment that is building towards the fork, as we see here, and one segment that's building away from the fork. So we have our leading strand, which is here, and then we have our lagging strand, which is here.
Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism.
So you can see in this video how the leading strand looks pretty easy. It's just building towards the fork. And this one here is a little bit more complicated. So it's kind of like pulling through the DNA and it's adding the nucleotide bases and then kind of coming back again to repulling that.
As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
All right. So the mechanisms of gene expression is that DNA and gene specifies information, but the information is not the structure and the function. So when I want a gene to turn on, the information, as we said, is specified in the information. So the genetic information expressed into the structure and function through what is known as protein synthesis. The expression of genetic information into structure and function is that DNA is a gene that determines the sequence of nucleotides in an RNA molecule. RNA then controls the primary structure of our protein. RNA then controls the primary structure of our protein. So DNA to DNA is replication. Now, the information from DNA has to come outside of the cell. So if I want my cell to do something and to make something, the information has to be transcribed and translated. So it transcribes DNA into mRNA. That process is called transcription. RNA then needs to be translated into something our body recognizes, and that would be our protein. Our proteins break down to amino acid sequences, and this protein here is then going to exhibit the trait. So RNA is a polymer of RNA nucleotides. RNA nucleotides contain the sugar ribose instead of deoxyribose. RNA's nucleotides contain uracil, adenine, cytosine, and guanine, or UACG. U is replaced by thymine in DNA. So there are no U's in DNA. The U's have been replaced, or U replaces the T in DNA. Sorry about that. There's 3 types of RNA which you should know about. We have messenger RNA, or mRNA, rRNA, which is ribosomal RNA, and tRNA, or transfer RNA. Okay? mRNA takes the message from DNA in the nucleus to the ribosomes in the cytoplasm. rRNA, or ribosomal RNA, makes up the ribosomes. And tRNA, or transfer RNA, transfers the appropriate amino acid to the ribosomes for protein synthesis. All right, so here's the difference between RNA and DNA. So the structure and difference between RNA and DNA. Okay, the genetic code of life. The unit of the genetic code consists of what is known as codons, and each of which is a unique arrangement of symbols. So there are 20 amino acids found in proteins, which is uniquely specified by one or more codon. This here is our codon chart, and this here is going to help us identify the amino acids based on the mRNA sequence. So transcription, let's get into transcription. So in transcription, we're going to go from DNA nucleic acid language to RNA nucleic acid language. Okay. So for this section, we're going to learn and be able to describe the mechanisms by which genetic flows or information flows from DNA to RNA to our protein. And like I said, these here are the essential standards from the AP College Board. You can read them on your own. All right, the first step in transcription. Okay, so let's say, for example, I drank milk and now my body needs to make the enzyme lactase. Lactase is the enzyme needed to break down milk. Now, the information to build lactase, which is a protein, is located in my DNA. Okay, so that information needs to be signaled. So signal is going to go inside of my nucleus, which will then tell my gene where lactase is located to be read. So transcription is the segment of DNA which serves as a template for the production of an RNA molecule. The gene unzips and exposes unpaired bases, and it serves as a template for mRNA formation. Now, loose RNA nucleotides bind to the exposed DNA bases using the CGAU rule. When the entire gene is transcribed into mRNA, the result forms a pre-mRNA transcript of the gene. The base sequence in the pre-mRNA transcript is complementary to the base sequence in DNA. So, to make mRNA, what does that mean? So, we're going to transcribe the DNA strand. That's called the template strand. So, like I said, I need lactase. So the information or the gene where lactase is located is going to open up. Then I'm going to go ahead and build the strand. So we have the template strand, and then we have the coding strand, which is the untranscribed DNA strand. From there, I'm going to synthesize the complementary RNA strand. And the enzyme responsible for building RNA is RNA polymerase. So let's say, for example, this here is the information to make lactase. This enzyme here is going to open up the strand, and I'm going to build my RNA. Now remember, DNA never leaves the nucleus, because if it leaves the nucleus, it can be taken over by a virus or bacteria. However, in order for the information to go out, it needs to be written in a code, or a code in which your cells recognize, which would be RNA. Okay. So here's another picture. Here's our DNA polymerase enzyme. It's going to add in our nucleotides, one base pair at a time. And it's going to build our RNA transcript. All right, which gene is read? So we have on our genes, which is known as a promoter region. So the promoter region is the binding site before the beginning of a gene. So like I said, here's our chromosome. Here's the gene of interest. So let's say this is lactase. Okay, right before lactase, there's this sequence, which is called our TATA box binding site. And this here is the binding site for the RNA polymerase and transcription factors. So it's going to be like a switch. And this switch here is going to turn on or off depending on if I need to make the gene or make the enzyme, in this case, lactase. So if I drink milk, now this enzyme here has to turn on so that I can read the DNA template strand, okay? The enhancement region or the enhancer region is the binding site for the upstream of the gene, and it turns transcription on high. Anytime we turn transcription on high, that means DNA to RNA. DNA to RNA is being made. All right, the initiation complex. Whenever I initiate, that's the start. So transcription factors bind to the promoter region. Once again, I drank milk. The signals now are going to attach on to this start region, okay? It will then turn on transcription and read the gene and build our RNA transcript, Okay, so it's going to trigger the binding of RNA polymerase to DNA. Now, eukaryotic genes have junk in them. So eukaryotic genes are not continuous. And that's basically a survival mechanism to make sure that we don't have errors, okay? We have these two terms that you'll need to know, exons and introns, exons and introns. Exons is our expressed or our coded DNA, the real gene, and introns is the junk gene, or it's in between sequence. It's a non-coding sequence, so codes for absolutely nothing, okay? So here's our eukaryotic DNA, right? Here's the information to make lactase. So we have coding and then our non-coding. So this is the expressed gene. This is the stuff that I need. This is the stuff that I don't need. Now, when mRNA reads the DNA strand, right, it's going to form a primary transcript, okay? So post-transcriptional processing. The eukaryotic mRNA needs work after it's been transcribed. So once again, once I read the DNA sequence, we then need to fix this transcript, okay, or pre-mRNA. And what we do is we are going to splice out the introns because all I want is the coding. In this instance, all I want are going to be these green segments here because these contain all the information to make the amino acids that I need. I don't need the orange stuff, or I don't need these introns, okay? So I'm going to splice out the introns and I'm going to put the exons together, okay, to make what is known as a mature mRNA transcript, And that's the transcript that we want to go out to the nucleus, okay? So the pre-mRNA is modified before leaving the eukaryotic nucleus, as we stated. Before it leaves, we need to add a cap, a 5' cap, and a poly A tail. And what that's going to do is, like we said, so if this here is my transcript, okay, so here's my mRNA, okay, it's already been done. Now, before it leaves, I'm going to add a cap, and then I'm going to add a tail, okay? What do these do? What these do is these caps and these tails is it's going to protect the code. Why? Because when the mRNA sequence leaves the nucleus, there's enzymes that are found on the cytoplasm, and those enzymes will eat away at these bases here. It's okay if it eats at these bases because they don't code for anything. We don't want it to eat at these bases here, okay? Because if it eats at those bases, then we can have mutations, okay? So once again, we add a 5' cap, and the cap is a modified guanine nucleotide. And then we add a poly A tail, which contains about 150 to 200 adenines at the end. So just A. And same thing, and it inhibits degradation of the mRNA by the enzymes found in the cytoplasm. So same thing, the pre-mRNA, just kind of regrouping. The mRNA is composed of exons and introns. The exons will be expressed. The introns will occur in between the exons and allows the cell to pick and choose which exons will go into the particular mRNA. Now, the introns are going to be spliced out and the exons will be put together. It's performed by what are known as spliceosomes, right? It just only makes sense. Spliceosomes are enzymes which take out the introns, okay? The result then is a mature mRNA transcript, All right, so here's a better illustration of our mRNA editing and processing. So don't forget, so here's our DNA, right? So here's our gene. Let's say, for example, here's lactase. We're going to build our RNA strand, right? Our original pre-mRNA strand contains our coded and our non-coded DNA sequence, or I'm sorry, our mRNA sequences. I add a cap and a tail. Then I have these enzymes, which are called spliceosomes, and they're going to splice out the non-coding introns and place together the exons, okay? So we have our mRNA, I have my cap and my tail, and then that's it. And then once our mRNA is done, it's going to leave and it's going to attach onto the ribosome. And our ribosome, as you guys remember, is where it reads the protein, and that's where proteins are made. Okay, you can watch this video on your own on the RNA splicing. All right, so in the next section, we're going to learn the description of how the phenotype of the organism is determined by its genotype using translation. So amino acids are coded by mRNA-based sequences, as we've seen. So once RNA is edited, it's now going to go ahead and it's going to leave the nucleus and attach onto a ribosome. So translation converts mRNA messages into polypeptides or our protein. That process is known as protein synthesis. So this is the process of making proteins using information coded on DNA and carried out by RNA. A codon is a sequence of 3 nucleotides that codes for an amino acid, as we see here. So every 3 letters on the mRNA strand code for a codon, and that codon gives us the information for the amino acid. Okay. The mRNA code travels from the nucleus to a ribosome. The mRNA is read in triplet codes called the codon, and the ribosome will build the amino acid chains to form a protein. So here's our amino acid, amino acid, amino acid. And as we see here, here is our RNA strand, and this large molecule here is our ribosome. All right, ribosomes consist of 2 subunits. The large subunit has 3 binding sites for tRNA, and the small subunit binds to mRNA. So this is the EPA sites, EP and A, and here is our ribosome. Here's our amino acid chart. Okay, to use this amino acid chart, I'm going to go ahead and break it up for you guys. All right, so let's say, for example, our RNA sequence is AUG CCA UAG. Okay, so what we're going to do is we're going to break it up into every 3 letters. 3, 3, and 3. So now I have 1, 2, 3 amino acids. So the first one is going to be AUG. So to do that, I'm going to start off with the very first A, which is in the middle, A. Then I'm going to go down to the next letter, which is U. So here's U, and then G. AUG stands for methionine. And when we write our amino acids, we are usually just going to write down the first 3 letters. The next one is CCA. So same thing, we're going to look for the start off in the middle. So we start off with C, C, A, and that is proline. So I have methionine, proline. And the last one is UAG, U, A, G. And that's our stop codon. So that means that we are done coding for your amino acid sequences. So we have the start and then we have the stop codon. A change in the order in which the codons are read changes the resulting protein. So if I switch, I then change the whole protein sequence, which can then cause our mutations. So regardless of the organism, codons code for the same amino acid. So same thing, we're going to use the amino acid table or the codon chart to find the sequences using information on our DNA strand. So here's our DNA, ATG, GTC, CGA. I like to kind of break them up already. So A goes with U, T goes with A, G goes with C, G goes with C, A, C, C. I'm sorry, G, C, and then A goes with U. Remember that there's no T's in RNA. Then I separate them out every 3 letters. UAC, CAG, GCU. We're going to go back to our codon chart. UAC is tyrosine, CAG is glutamine, and GCU is alanine. Okay. So like we said, the amino acids are linked to become a protein. And if you recall, the amino acids make up our proteins. So an anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. Okay? An anticodon is carried out by tRNA. So an anticodon is carried out by tRNA. So here's our tRNA molecule. It has the anticodon, and it also has our amino acids. So identify the anticodons for the following codon strand. Now, in order for the amino acid to basically be read, the tRNA molecule is going to attach to the mRNA strand using base pair rules. So it's RNA to RNA, same thing. There is no T's. We're done with T's. That's only in DNA. So these here will bind. CAG will bind with GUC. ACC will bond with UGG, and so forth. So for translation to begin, the tRNA binds to start codon, and then it signals the ribosome to assemble. So a complementary tRNA molecule binds to the exposed codon, bringing its amino acid close to the first amino acid sequence. So in translation, we convert the mRNA into a polypeptide, okay? So here's our RNA strand. Like we said, it's going to attach onto our ribosome. tRNA is going to come in here, and it's going to attach, as we see, to the 3 letters, to our codon, okay? And then here is our anticodon, and here's our amino acids. Next thing you know, the next tRNA is going to come in, carries the anticodon, attaches to the codon here. These 2 will then link together. And if you guys remember, let's see how great your memory is. What molecule gets released whenever I put 2 molecules together? That process is called dehydration synthesis. I'm hoping you said water. So water would be released here. Water would be released here. Water would be released here. And it's going to keep continuously bringing on these tRNA molecules, tRNA, and it's going to read until it reaches the stop codon, So like we said, the ribosome helps form the polypeptide bond between the amino acids. The ribosome pulls the mRNA strand length of 1 codon. Now the empty tRNA molecule exits the ribosome, and then the complementary tRNA molecule binds to the next exposed codon. So one's leaving, one's holding, and one's entering, as we see here. So this one here is leaving, this one's staying, and this tRNA is going to enter. So that's our EPA site. So EPA. And notice here, these are all our amino acids. Eventually, they're going to form our protein. Once our protein is once we are done, our stop codon will indicate our UAG, and then the protein gets released. Now, let's see if you guys remember from the ER, okay, from the ER, where does the protein go? So from the ER, it's then going to go to the Golgi, and it goes out. All right, so once again, mRNA to protein is called translation. The reader is the ribosome. The transporter is our tRNA molecule. All right, so finally, you probably forward or went ahead. Hopefully you guys pay attention to this one. This is everything that you should know for transcription, translation, and protein synthesis. So once again, I drink milk, My body's going to send a signal for the gene to open up where lactase is located. Okay. RNA then is going to be built. I have my pre-mRNA transcript, okay, to make our lactase enzyme. Introns get spliced out, exons get put together. I add my cap and my tau to form my mature mRNA transcript. From there, my mature mRNA is going to transfer out of the nucleus and attach onto a ribosome, okay? Once it's on the ribosome is going to read the mRNA in every 3 letters known as our codon. The tRNA molecule is going to bring the amino acids to our ribosome where it's going to attach to the codon, anticodon, codon. Base pair rules here. Then another tRNA molecule will come in, and as we see here, one tRNA molecule is leaving, one is staying, and then the next one is entering to now we will build, and then it's going to continue and keep moving down the line till it keeps reading, all the way up until it reaches a stop codon. Once that stop codon is done, our protein is built as we see here. Protein that will then go from the ER to the Golgi, and then from the Golgi out. Okay. All right. So I'm hoping that you guys remember this story. So DNA to RNA is transcription, RNA to protein is translation. Finally, just in case you guys forgot, you need to know the structure of the chromosome. I know we did this in mitosis, but please make sure that you recall. Please tell me the structure of a chromosome. So remember, if I were to untangle a chromosome, it's made up of a really long linear strand of DNA, okay? But it's also comprised of 50% protein. So DNA is going to wrap around these proteins, which are called histones, okay, histones, and it helps for packaging up the DNA. So once again, here's our chromosome. Remember, I untangle it. It's a really long strand of DNA, and it's packed with these molecules or these proteins called histones. All right, I know this is a really long lecture. Thank you for staying here. If you need to go back and forth to review, please do so as needed, and I hope you all have a fantastic day.